All right, I'm gonna give it one more minute and then we'll get started. Folks, like I said, folks are unusually late today. But go ahead and log into Canvas. I'm not going to follow it exactly because they hinted some things and then move around. Okay, for some reason, every time I share, I lose you guys. I don't know why that is. That is very strange. Okay, so in Canvas, I want you to be here on the page. I'm going to skip her. I find her enthusiasm a little bit annoying, and she starts in a very unusual place. So we are going to be talking about cellular reproduction, and we're going to start it on a cellular level. So we need to talk about cell division. So I do want to start with this one, and then we're going to look at, she's going to bring up a topic of the cell cycle. And then we're going to skip to mitosis and then skip down to this summary, skip her again, to this summary down here. So those are the important things that we're going to cover today. So I'm going to go ahead and show this one. And then from there, we will look at the Nearpod that talks about the cell cycle.
Before we start, let's begin by reviewing DNA. DNA stands for deoxyribonucleic acid. It's a type of nucleic acid, which is one of the 4 macromolecule groups. When a eukaryotic cell is not reproducing, its DNA is inside the nucleus as disorganized long strands called chromatin. Before a cell divides, the chromatin thickens and shortens into distinctly visible bodies called chromosomes. Each chromosome is a single DNA molecule. In order for a cell to divide, its chromosomes must first make a copy of themselves. This is DNA replication, so that each new daughter cell gets the original amount of chromosomes. Just after chromosome replication occurs, the 2 chromosomes, old one and new one, are stuck together. These 2 structures are referred to as sister chromatids. They are joined together by a central protein bundle called centromere. The sister chromatids are exact copies of each other, and each one will be distributed to one of the new daughter cells. Now that we have reviewed DNA, let's discuss mitosis.
All right, so I want to stop there for a minute. First of all, she said DNA really weird. It's deoxyribonucleic acid. And we are looking at eukaryotic cells, which are the cells that have membrane-bound organelles. So we had prokaryotic cells and eukaryotic cells. And that was something we talked about way back at the beginning of class. And so we are looking at what eukaryotic cells do. Okay. And so We're particularly only going to be looking at what's going on inside the nucleus, which is where the DNA is housed. So for the rest of the unit, the only structure that we're really talking about is what's happening in the nucleus. Everything we're going to talk about mitosis and meiosis, and basically it's going to be about what is happening with the DNA and what happens to the nucleus, and then eventually they'll just generally say, and the rest of the cell divides. But most of what you have to know is what is the chromosomes, what are the chromosomes doing, and what are the phases of mitosis and meiosis. All right, so she's going to give you a quick uh, overview, but then we're going to break it down into individual phases.
Cell division is the production of creating new cells. Cells divide in order to grow or repair damaged cells. An example is your skin or intestinal lining, which divide regularly to grow in size and or repair tissues. Not all cells divide though. A number of cells in your body, such as nerve, red blood, and muscle cells, do not divide once they are mature. The cell cycle is a timeline of events that occur during the life of a cell. The cell cycle is composed of mitosis and cytokinesis. The cell spends most of its time in interphase, which is the green area. During interphase, the cell is getting ready to divide. Interphase is divided into three sections, G1, S, and G2. In G1, the proteins are produced for the cell that will be necessary for cell division. In S, DNA makes a copy of itself, which is the DNA replication. In G2, cells make copies of the organelles. So mitosis is a type of cell reproduction that produces identical cells. This means producing cells that have the same DNA.
All right, so our basic goal is to produce cells that have the same DNA. So, and that's going to be our difference between mitosis and meiosis. Mitosis is going to produce identical cells, and the next one is not. So we'll cover that as we go through it. Somebody said they couldn't find the unit that we were on. It's the CR mitosis lesson. And again, we're going to skip Jolly Lady here. And so that's an overview. So they talked about cell cycle. So what we're going to do is we're going to do a Nearpod, and I'm going to send you. So I'm going to stop sharing for a minute, send you the link, do attendance, and then come back and go through this with you. It's not there. It should have been. It's not published. It's published. Hold on. Let me come out. Sometimes I publish individual modules and not the whole thing. And it should be. Give me 1 second. I do believe that that is the problem. Okay. And had y'all looked at this yesterday, you could have told me this yesterday because I asked y'all to look at this yesterday during thing. Okay. It's published now. Okay, so if that's the case, you all need to go back because I showed this to you, but I guess if it wasn't published, you didn't do it. All right, so while I'm getting the link for the, go back to the first page. It says cell reproduction and look at the overview. There are a lot of vocabulary words there. So you need to look at that page first. And just give yourself an overview. of all the vocabulary that you need to know for this unit. Okay. And then I am sending you the link now. See, sometimes y'all tell on yourselves. All right, so that's the link for the Nearpod. And I did not send it as student mode because I want to go through it with you. All right, so one of the things she talked about was the cell cycle. So once everybody has logged into that, I will. Okay. It wasn't an assignment. It was just, you were just told to look at the vocabulary after the test. And like I said, I looked at the page that had the vocabulary on it. It said publish, but what happened, the way Canvas is, you have to publish the whole unit before you can publish individual modules. So while the modules were published, the unit wasn't published. It happens. I'm human. Thank you very much. All right, let me get this attend. All right, so while I'm doing attendance, make sure that you are looking at everybody should be logged into the Nearpod right now. So right now I'm looking at Go Guardian, and everybody ain't there yet. I got 2 people absent, so let me mark them absent and we'll go on. Okay. I think Michaela is coming, but she's not there yet. There she is, which means I've got to post the link again. Okay. Okay. All right. In any case, everybody should be on. Oh, we got one more coming. Sometime today. All right, I'm going to go ahead and oops, that didn't go to everybody, did it? I'm waiting for her to get in before I post this again. All right, in any case, everybody should be on the Nearpod page. Everybody. All right, I'm gonna go get started because she's not logging in and she's just late. Okay. Okay, so we are looking at the stages of the cell cycle. And I'm going to want you to write this. So you're going to have to draw a nice, take out a sheet of paper, draw a nice big circle, and we're going to label the stages of the cell cycle. All right, so before we get to that part, there's a to-do activity. So there's only a couple of things that they want you to label. So take a few minutes to label this diagram. Let me see how many of y'all. So everybody should be trying to label this diagram. Now notice they're not asking you to label everything because like I said earlier, we're only concerned with certain activities that are going to be involved in cellular reproduction, and that's not everything in the cell. So there's 4 things that we're all right. Okay, folks, everybody should be trying to label the diagram. Let's go. Oh, finally, she made it. Okay, so Michaela and Keela, the link for the Nearpod has just been entered so that you can join us quickly. All right, so there are 4 structures that they want you to label. I want to pay particular attention to the fact that there is a large central object and there's something inside the object. There's our little energy organelle and some other little dots floating around the cell. So take another minute to answer those. Okay. Water, nutrients, no. But you're right about the rest. All right. So I pulled Jacobs up because he's got them labeled correctly. All right. So the large structure is the nucleus. The smaller structure is inside the nucleus. It is the nucleolus. I'm not sure you spelled everything correctly. The red structure is the mitochondria because remember, we need energy for everything. And these little dots that are floating around are ribosomes. Those are ribosomes. So go ahead and correct that, please. All right, Jacoby, we're waiting. Everybody's got to participate. Remember, this gives me a record of who participates and who don't. And since I just gave you all the answers, everybody should be fixing their diagrams accordingly. All right, so while you're doing that, so we are doing cellular reproduction, and this happens when we say in eukaryotic cells, this is going to happen in plants and animals both. We'll look at the difference of what happens in plants, but plants are going to do the same inner procedure that the animal cells do as well. Okay. All right, I'm gonna go on because some of y'all are deliberately dragging your feet, but that's okay. Like I said, it'll show up on participation. All right, we're going to explore. All right, so we're going to look at the cell cycle. So I asked you to take out a sheet of paper and actually we're not going to do this piece because it asks you to do it before they even give you, whoops. Okay, hold on a second. It skipped something. Maybe it didn't. Yeah, it did skip something because we're not really trying to do that. All right. There should have been a piece for you to draw. So hold on 1 second. Let me come out of this. Sorry about that. Okay, hold on. Really want to get back to, stop sharing for a minute. I tell you what I'm going to do. And I picked this one because it had the cell cycle drawn. Mm-hmm. Okay, it doesn't. Okay, what? Because they went on to ask you questions that.
Hi there. Let's discuss.
Okay, I'm going to go back to, come out of the Nearpod and go back to Canvas. And then I'm going to, because they took you to a site that it opened for me earlier, but I guess it's not going to open for you. I don't know why they put it in there if it's not going to open for you, but they did. All right, so come back to Canvas. I'm on the video that I showed earlier, but I'm going to pause it and enlarge it so that you can draw the cell cycle. So put a title on your page, call it cell cycle, and you have to have it labeled the way she has it labeled here.
Life of a cell.
Okay, so I'm going to leave it right here for a minute. So the cell cycle is like a time clock of what the cell does during cell division. So so you need to know what cytokinesis is. You need to know what interphase is and what is happening during G1 synthesis and G2. And then, so it's going to go in a clockwise fashion. And cytokinesis simply means that the cell divides in two. Okay, cyto is cell, kinesis is division. So cell division. And we're going to look at, and we're going to learn that the initials for the phases of mitosis are PMAT. So I do want you to draw it just like this. So basically you're drawing two circles, a circle inside a circle. You're going to label it cell cycle. And we are going to spend a lot of time in just this little red section over here. But you need to understand that when it's not doing that little red section over there, it is an interphase. So the majority of what this clock is showing you is the majority of the time, the cell is just getting ready for cell division. It's prepping, it's growing, it's doing all the things cells do until it's time to divide. So you might even add an arrow, like that kind of goes around to show you that it's going to go in a clockwise direction. So again, you have a circle inside a circle. And the near part, the part that it was taking you to, is going to take you to a website that actually had this drawn and labeled for you, but it wasn't going. So doing the following questions wasn't going to help you. And I logged into it earlier before you came in, but apparently this time you need permission. All right, so I'm going to move it up just a little bit. And I'm going to let it talk.
The cycle is composed of mitosis and cytokinesis. The cell spends most of its time in interphase, which is the green area. During interphase, the cell is getting ready to divide.
Okay, so I want you to write that. I want you to underneath, see the word down here at the bottom, interphase. I want you to say what it says down there at the bottom of the screen. During interphase, the cell is getting ready to divide. So that's most of the cell cycle is interphase, and you need to know that. And then later we'll talk about. So when we say, what are the parts of cell division? Interphase is really not one of the parts. So it's not going to be part of the initials of PMAT. All right, so all you had to do was write the word interphase and then write during interphase, the cell is getting ready to divide. Okay.
Interphase is divided into three sections, G1, S, and G2.
Okay, now write that down. interphase is divided into 3 sections so that you should have that already labeled on your circle.
G1, the proteins are produced for the cell that will be necessary.
You don't have to
for
write
cell
all
division.
this.
In S, DNA makes a copy of itself, which is the DNA replication. In G2, cells make copies of the organelles. So mitosis is a type of cell reproduction that produces identical cells. This means producing cells that have the same DNA.
Okay, so now we're going to look at mitosis. Okay, so now I'm going to share, because again, she's a little wacky. And basically I'm going to give you the visual on this process right here. All right, so give me 1 sec to get this one going. So mitosis is that one section that's not green on your cell cycle, and that's what they're going to talk about here. And actually, I'd like
Have
to
you ever looked down at your leg or your arm to find some cut and you have no idea where it came from or how you got it? So you put a bandage on it and a few days later it's gone. It's all healed and you don't even think anything of it, of this amazing process that causes this to happen. Or let's say you're looking at your nails and you notice they're a lot longer since the last time you cut them. Or let's say you're looking in the mirror and you notice that you are a lot bigger than you were when you were 5 years old. What do all of these things have in common? Well, one major thing they have in common is mitosis. Mitosis is a type of cell division done by most of your body's cells, and it's really important for cells to divide.
All right, so that's the first thing you need to write down. You don't have to write all of this. Mitosis is done by body cells. And that's important to remember because when we do meiosis, they're done only in the reproductive cell. So all of your body cells, and even with the plant, all of the plant structural cells are going to do mitosis in order to carry out cellular division. Okay, it's done in the body cells. That's all you have to write.
If they didn't divide, you wouldn't grow. I mean, how do you grow if you can't make more cells, right? So one reason why you're bigger than you were when you were 5 is mitosis. Mitosis also is great for repair of damage. If you have some kind of accident, like when we were talking about that cut on your arm or leg, well, you want to make sure it gets repaired well. So you have to make more cells to do that. Mitosis is great for that. Now, it's really important to understand what it is not. Mitosis is not a process that makes sperm or egg cells because that's something different called meiosis, which sounds like mitosis, unfortunately, but it's a different process. Mitosis is done to produce body cells. Mitosis makes identical cells. That is the goal, identical.
All right, write that down also. It makes identical cells, which means all of the chromosomes are going to look like all of the new chromosomes.
Cells. So if you're trying to make more skin cells to replace worn out or damaged skin cells, you don't want to start suddenly making stomach cells there. That would be ridiculous. You want to make sure you have identical cells replacing what was lost. So mitosis makes identical cells. It's a really important thing. Now, it's also important to understand that your cells are not dividing all the time. If all they did was divide, it would just be rapid, crazy growth. In fact, this is kind of what cancer is. Cancer is uncontrolled cell growth. We have a clip on the cell cycle and what the cell is usually doing most of its daily life, which is actually a phase called interphase, where it's growing and replicating its DNA and carrying out its daily cell functions. That's where
Okay, you can write that under interphase. Interphase has growth, DNA replication, and if I can just take the closed caption off for a minute, cell function. So that was what your G1, your S, and G2 were about. In fact, you could write that next to it, G1, S, and G2. All right, so also remember what she said too. If it was reproducing all the time, cancer is simply uncontrolled cell growth, and cells are just growing on top of themselves and growing in places where they shouldn't grow. And that's all cancer is. So unfortunately, growing like that causes complications. It's not a good thing.
where cells spend most of their time in respect to the whole cell cycle. Mitosis is a very short amount of time in respect to the whole cell cycle. But mitosis is a critical process because this is where it is going to divide and make more cells. Before we get into the steps of division, it's really important to understand that your cells have something inside them, an organelle called the nucleus. And the nucleus holds your DNA. DNA is really important because it's your genetic information. And if you're going to make more cells, you need to have the same DNA in those new cells as you did in your original cells. You want it to be identical, no mistakes. Very important. The problem is you've got a lot of DNA, and we've got to get that DNA into the new cells using mitosis. So there has to be a better way to organize that DNA. Well, what actually happens is that DNA can be organized into these condensed units called chromosomes. Chromosomes are made of DNA and protein. You've probably heard before.
Okay, write that down. Chromosomes are condensed DNA and protein. Chromosomes are condensed, and then it's not written there, but make sure you write that down because originally it looked like spaghetti. And then when it's time for cell division, they condense, which means they shrink, they coil up really tight and form the structures called chromosomes.
That humans have 46 chromosomes. That means 46 chromosomes are found in most human body cell nuclei. What are nuclei? Well, it's the plural of nucleus. You don't say nucleuses, you say nuclei. Well, in the nuclei, there are 46 chromosomes. Organizing DNA into condensed chromosomes makes it a lot easier to move over when you're making new cells. So if you have 46 chromosomes in a human body cell, you have to duplicate those chromosomes in interphase before mitosis starts. That basically means you're duplicating your DNA since chromosomes are made of DNA and protein. You have to do this before mitosis starts because if you're going to make an identical cell that has 46 chromosomes just like the original, well, it makes sense you have to duplicate the genetic material before splitting. So if you look at our cell cycle video clip, we talk about interphase. That's a stage where most of the time cells are spending their time. They're actually duplicating their DNA during that time. So ready for the tricky part? Well, because we tend to count chromosomes by the number of centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes, as the sister chromatids are still attached. And we're counting by centromeres. So 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture, but you went from 46 to 92 chromatids. We have a video explaining that in more depth and how that factors in for mitosis. Okay, so now we can get right into mitosis. I like to tell students to remember PMAT. It's a little acronym that helps you remember. The P is for prophase, the M is for metaphase, the A is for anaphase, and the T is for telophase. So remember PMAT, the stages in order. The very first step is prophase. Prophase because it's the beginning step, the nucleus is still there. And it's going to go away later on, but this is a stage where it's actually still there. The chromosomes are visible. In fact, we say they're condensing, which means they are thickening and visible. The next stage is metaphase. M for metaphase, but I also like to remember M for middle because in this stage, the chromosomes line up in the middle of the cell. The nucleus has been disassembled, it's no longer there, so we've got the chromosomes in the middle waiting there. Next, the A is for anaphase. In anaphase, I like to think as the A for away. The chromosomes move away. They are moving to opposite sides of the cells. They are moving towards the poles of the cell. Now, one thing to point out, these chromosomes, they're not moving by themselves. They actually have something called spindles. These spindles are fibers that help move the chromosomes to the ends, kind of helps it move it on. The last stage of mitosis, think T for telophase. In telophase, the chromosomes are actually at the complete opposite end, and new nuclei are forming on each side to make two new cells. The nuclei are starting to surround the chromosomes on both sides. I like to think of the T42 because you can really see that the end goal is going to be two cells. And in the human body, they're each going to have 46 chromosomes. And again, remember, they are identical. And cytokinesis is responsible for the final separation into two cells by splitting the cytoplasm, which completes after the PMAT mitosis stages.
I'm going to go over it again when she's finished.
Without understanding cell division, we wouldn't understand how growth and repair happens, because they both require more cells to be made. Understanding mitosis is also very important for cancer research.
All right, a couple of folks saying the sound is lagging, so I'm going to stop it there, and we're going to go back and look at the pictures in Canvas. All right, so she went through what the sisters went through. All right, so they've got two things here listed for prophase. So we have prophase and prometaphase, but this is the part that you need to focus on. So again, remember PMAT because you do need to know the stages in order. You also need to be able to look at a picture of the cell and understand what is happening in that picture and be able to label it. All right, so prophase. So we had interphase and we've made copies. So the cell has gotten ready for division. And so and then she also mentioned 46 chromosomes. All organisms have a unique number of chromosomes. Humans have 46. Not everything has 46. Plants have different numbers of chromosomes. That's how you can look at a cell, count the chromosomes, and identify what it is by counting the chromosomes, because every organism has a unique number of chromosomes. All right, so for humans, it's 46. So we take those during interphase, those 46 chromosomes are making copies of themselves, okay? And that was during the S phase, the synthesis stage. They're making copies of themselves. When they do that, the copies line up, they kind of hang on to each other, and then we're in prophase. Okay, prophase is like the getting ready stage. So the nuclear membrane is still around the chromosomes. The spindle fibers are starting to form. So everything is getting ready during prophase. Okay, the chromosomes are condensed, spindle fibers are formed. And the nuclear envelope starts to break down. So when you're going to see it, it looks like that little dotted line around the orange section. That's the nucleus is still there, but it's starting to break up. And the centrioles move towards the poles. So the spindle fibers come from the centrioles. The job of the centrioles is to create these little fibers. And while they're together, then they start to move apart, like we call it maybe the north and south side of the cell. And then by the time this next one, the prometaphase, then they're already lined up on either side of the cell. So it says it continues to get ready, and the spindle fibers start to grow, and then they're going to attach themselves to the chromosomes. Okay, remember I said the pairs are hooked together, the copies are hooked together. All right, so what it's doing is the south side is going to hook up to one copy, and the north side is going to hook up to the other copy. In metaphase, and this, like she said, think of metaphase as the middle. The chrome, you know it's metaphase, there's no nuclear membrane, it's totally gone. And the chromosomes are lined up in the middle, and the spindle fibers are attached. Okay, so before that, they were getting ready, they were getting all the preparation done. Now they're lined up in the middle, and spindle fibers are attached. Next one, anaphase. What those spindle fibers are doing now is they're going to pull those copies apart. Okay, so like she said, we had 46, now we've got copies. So there's 90, we had 92, got 92 chromosomes. And when we pull them apart, each set is going to be 46 and 46. Okay, so, and there's still no nucleus there. All they have, and like she said, you can think of the A for away. You're pulling them away, the copies away from each other. And the spindle fibers do that by shrinking. All right, telophase is the last phase of mitosis. And what happens there is the nuclear membrane starts to form around each of those sets again. The spindle fibers totally shrink and become detached from the chromosomes. And then they start looking like chromosomes again instead of the little short stubs that they were. And that's the last stage of mitosis. What happens after that is cytokinesis. And then in cytokinesis, then everything else divides. The rest of the cell divides. The cytoplasm and the cell membranes pinch in, and that's where you form the two new cells. Okay, so you need to know these in order. You need to know the description of each of these. I would say I'm going to give you next class a worksheet that has the pictures already on them for you, but I'm going to expect you to be able to fill in what is happening in each phase of mitosis. So again, remember PMAT, and PMAT sits in the middle of interphase. Interphase takes up 90% of the cycle. And that's where it's getting ready to divide, and mitosis is the process of cell division. Okay, so take a few minutes to look at that, reread each one, look at the picture, see what's happening. Because again, you do need to know what is happening in each phase. You do need to be able to recognize each phase. If I mix them all up, you need to be able to put them back in order. All right, so after you've done that, all right, so do they reproduce the same? Like I said earlier, the steps for mitosis are the same, except with plant cells, because they have a cell wall. When they get ready to divide the cytokinesis part, they actually have to create a cell wall plate first that divides the two cells. But other than that, it goes through PMAT the same way an animal cell does. And plant cells don't have centrioles, but they do have microtubules, which act like centrioles that are still going to be involved in pulling the chromosomes apart. All right, take a minute to go through this drop and slide section. So you're going to take okay, how are we doing this? It says drag and drop, but mine isn't dragging. There we go. Okay, so you got two. Mm. All right. So if you notice, it won't stay. If it's not right, it won't stay. All right. So you should be able to do a box like this, and know what's going on during each phase. And you should be able to do it by looking at the pictures above. So relate what's happening in each of these boxes with what is happening in the pictures. All right, so in summary, you need to know there are two phases of the cell cycle: interphase, where it's getting ready to divide, and mitosis, which is when it is dividing. You need to know, and again, most don't deal with pro-metaphase. They simply do prophase, metaphase, anaphase, and telophase, but they're stretching out the prophase part, but that's the getting ready part. This is the lining up in the middle, pulling apart the chromosomes, and then creating two nuclear sets of chromosomes, and then the cell divides. Okay. All right. Any questions about that? So again, when I see you, because there's no school on Monday, and so I won't see you till Wednesday. So I'll have a little activity where y'all will have to match these up. So make sure that you're studying that. All right, so the next one, so we're going to take a minute to look at that. All right, so the next one is meiosis, and that's not published for you yet. But we'll do that after. And then we're going to compare what is happening. You need to be able to say what's the difference between mitosis and meiosis. So it gets a little challenging. All right, so I'm going to leave it there. If you have no questions for me, we're good. So have a good weekend and see you Wednesday. That's what she was.